Welcome, everyone, to The Explainer. I am absolutely thrilled to have you here today because we are looking at a technology that is completely revolutionizing how we study the brain in motion. It's called Functional Near-Infrared Spectroscopy, or fNIRS. If you've ever wondered how we can actually peek inside the human mind while someone is, walking down the street, having a conversation, or even playing the piano, well, you are in the exact right place. Let's get into it.
So for decades, neuroscientists have faced this massive roadblock. Think about traditional brain scanners for a second, like fMRI. They're incredible, right? But they require rigid stillness. You literally have to lie perfectly flat inside a loud, narrow tube, and oh yeah, the machines cost millions of dollars. Because of this, our understanding of neuroscience has historically been trapped in highly artificial environments. We could study the brain, sure, but not the brain out in the real world. So how do we fix that? How do we map the mind in natural motion?
Here is our roadmap for today. We'll be covering 1. The neuroimaging revolution. 2. Hemodynamic and metabolic signals. 3. fNIRS workflow step by step. 4. Clinical and cognitive frontiers. And finally, 5. Inclusive design in neuroimaging. All right, let's jump straight into section 1, the neuroimaging revolution, finding the sweet spot. Okay, let's break down this definition. First demonstrated by Franz Jopsis back in 1977, fNIRS is a completely non-invasive optical technique. The real magic here lies in what we call the optical window. These are light wavelengths right between 600 and 1,000 nanometers. In this very specific range, our biological tissue, like water, lipids, and bone, is actually relatively transparent. This allows near-infrared light photons to travel right through the scalp and skull, dip down into the superficial layers of the cerebral cortex in this distinct banana-shaped trajectory, and then bounce right back out to a detector. I mean, we're literally shining light through the skull to see what the brain is doing. How cool is that? To put things into perspective, let's compare it. Look, fMRI gives you gorgeous high spatial resolution. You can see exactly where things are happening, but you have zero portability. Now, on the other end, EEG gives you blazing fast temporal resolution. You see exactly when things happen, but honestly, it's pretty terrible at telling you where. fNIR sits perfectly right in the sweet spot. It offers moderate spatial and temporal resolution, sure, but it is highly wearable and highly resistant to physical movement. It completely bridges the gap. Moving right along to section 2, hemodynamic and metabolic signals, the language of light. Now, here's the kicker. fNIRS doesn't actually measure electrical neurons firing directly. Instead, it relies on something called neurovascular coupling. When a specific region of your brain starts working really hard, say to solve a math problem or move your hand, those neurons demand more oxygen and glucose. Your cerebrovascular system instantly responds by rushing a localized oversupply of oxygenated blood to that exact area. We call this functional hyperemia. And that rush of blood, that is exactly what fNIRS is detecting. Traditionally, fNIRS uses just a couple of light wavelengths to track both oxygenated and deoxygenated hemoglobin, relying on mathematical formulas like the modified Beer-Lambert law. But the real new frontier here is measuring cytochrome C oxidase, or CCO. Think of CCO as the literal engine inside your cell's mitochondria. I mean, it's responsible for 95% of cellular oxygen use. It's a much deeper, more direct brain-specific signal than just looking at hemoglobin. Detecting it, though, well, that's incredibly tough. It requires advanced broadband NIRS using over 100 different light wavelengths just to isolate the signal. Depending on what exactly you are trying to measure, engineers have developed 3 main architectures for fNIRS systems. Continuous wave is the most common one. It simply measures changes in light intensity, which makes it absolutely perfect for cheap, simple wearables. Frequency domain uses modulated light to measure phase shifts, giving you absolute concentrations of blood oxygen. But time domain? That is the absolute pinnacle. It fires ultra-short picosecond pulses of light and uses a literal stopwatch, something called time-correlated single photon counting, to measure the exact time of flight of the individual photons. This lets scientists completely decouple light absorption from scattering, which gives them unparalleled depth specificity. Let's move to section 3, fNIRS workflow, step by step. So how do you actually use this stuff? It's a highly structured 4-step journey. First, you start by placing your light emitters and detectors, which we call optodes, usually following the standard 10-20 system that's also used in EEG. Next, you check your scalp coupling to ensure you have really good physical contact. Then, you capture the signals, making absolutely sure you include short separation channels. And finally, you perform artifact mitigation. Let me tell you, Pristine data collection here is essentially a detective story. You are constantly trying to isolate the true neural signal from just an overwhelming amount of bodily noise. And speaking of noise, here is the problem. Because the near-infrared light has to pass through the skin and scalp twice, once on the way in and once on the way out, it picks up absolutely everything happening in the superficial layers. It picks up the rhythmic pulsing of your heartbeat, it picks up the rise and fall of your breathing, it even picks up these low-frequency blood pressure changes called Mayer waves. Honestly, if a participant just gets a little stressed by the task you give them, their heart rate spikes, and that alone can completely mask the actual brain activity. To fight this, researchers use two seriously brilliant techniques. First is the Scalp Coupling Index, or SCI. They monitor the heartbeat waveform in real time, and if the SCI is greater than 0.8, they know for a fact they have a solid optical connection. Second, and this is the really important one, they use short separation channels. Imagine placing a detector less than 10 millimeters away from a light source. Because they are so incredibly close together, the light doesn't actually penetrate deep enough to hit the brain at all. It only measures the scalp. Using software like Homer 3, researchers can then mathematically subtract that pure scalp noise right out from their deeper brain recordings. It's just such an elegant solution. And when you get that processing right, the results are staggering: 4.4 millimeters. That is the reduced localization error achieved by the newest very high-density diffuse optical tomography systems. By packing the optodes incredibly close together, under 10 millimeters apart, fNIRS can now reconstruct 3D maps of brain activity with a spatial fidelity that absolutely rivals fMRI. Think about that for a second. We are talking about precision narrower than the folds of the brain itself.
All right, Section 4, Clinical and Cognitive Frontiers.
I absolutely love this quote from a literature. fNIRS enables the neuroscience of the everyday world. We have officially left the laboratory, folks. We can now actually monitor the brain while two people are having a natural conversation, while an elderly patient is navigating a walking test, or even while a pilot is flying a simulator. The applications here are wild. In the neonatal intensive care unit, fNIRS is a total game changer. An infant's skull is so thin that the light penetrates super deeply, allowing doctors to monitor mitochondrial oxygenation in real time for babies who might be suffering from oxygen deprivation. For stroke recovery, therapists use it to actively track how the premotor cortex reconnects to the primary motor cortex during physical therapy. In psychiatry, it's emerging as an objective biomarker. And in brain-computer interfaces, by using convolutional neural networks, fNIRS can decode what specific movement a completely paralyzed patient is imagining with 90% accuracy. That literally allows them to communicate with the outside world again. Which brings us to our final section, Section 5, Inclusive Design in Neural Imaging. We have to talk about a really critical issue here. The reality reported across the source material is that conventional fNIRS hardware was simply not built for all heads. There are strict biophysical barriers to signal quality. First up, skin pigmentation. Melanin heavily absorbs near-infrared light, meaning traditional calculations often fail to accurately measure oxygenation if someone has a skin tone darker than a 2 on the Fitzpatrick scale. Second, dark hair actually absorbs significantly more light than light hair. And third, hair texture. Traditional swim cap-style fNIRS nets mechanically fail on thick, coarse, or curly protective hairstyles, literally blocking the optodes from touching the scalp. Historically, this has led to the really unfortunate exclusion of diverse populations from vital neuroresearch. But the good news? The field is actively engineering solutions right now, and inclusive best practices are finally becoming the standard. Engineers are developing active, spring-loaded optodes on these flexible plastic mounts that can actually weave through protective hairstyles to establish direct optical contact. Furthermore, it is becoming completely mandatory for researchers to report phenotypic metadata tracking skin tone and hair texture to ensure total transparency and to push the technology to work accurately for absolutely all populations, rather than just a select few. Which leaves us with this final thought. We are watching a powerful technology evolve right in front of us in real time. The hardware is shrinking down into everyday wireless headsets, and AI is automating the incredibly complex data analysis. We are entering an era where high-definition brain imaging is accessible, portable, and increasingly inclusive. So whether you are interested in clinical diagnostics, sports science, or just the fundamental nature of human thought, what mysteries of the embodied human mind will you decode next? Thank you so much for joining me on this explainer. Keep learning, and I'll catch you next time.